So anyone who knows me knows I'm a complete proteomics homer, and so it is with a lot of enthusiasm that I'd like to introduce Henry Rodriguez. I tried and failed to add a slide to the slide deck a moment ago, but the picture is one that I got from Javad, who's leaving, of of a caterpillar and a butterfly in the same photograph. And the point that it makes is that those two organisms have the exact same genome, but they have different proteomes. And I think it very eloquently shows that the cellular phenotype is dictated by the proteome much more so than other layers, although all are important. And so with that thought, I'd like to introduce Henry Rodriguez, who is the lead of the NCI's CPTAC program, which he'll describe to us. CPTAC has really emerged as one of the most generous and influential collaborations that CPTN has. They have really stepped up into the pediatric realm. This marriage, if you will, has started with our access to high quality tissue specimens. That is the grist for the mill for the CPTAC machine, and it has really just been incredibly fruitful. And we're so very excited that Kids First has stepped up to support this work as well through the XO1 mechanism. And CPTAC has added additional resources and analytical capabilities. And so it's really been a dream to work with Henry and his group. It's a pleasure to introduce him.
First of all, let me just say thank you very much to Adam and also to Brian for the generous invite to the CBTN. So what I'm going to do actually is, I love the presentations today. Apologies that I could not come yesterday. If people saw, there's stuff going on in Florida where I have a lot of family right now. So, but that said though, my talk, I'm going to give a quick little overview on what I would call how to be inclusive. Inclusive at the research level and inclusive at the collaborative level. So what exactly do I mean by that? So in terms of the research, for all transparency, my background is actually in genomics. But when I ended up training in California, it was more on the clinical side, specifically trials and drug development, which is where our office is now currently housed at within the National Cancer Institute. So this is what I mean by inclusivity. I still remember when I learned biochemistry, so it goes from genomics to proteomics. There's a DNA, RNA, and a protein. But I get it. From a technology perspective, 20 years ago, one methodology was a horse that quickly outran the other one. So what we ended up doing at the National Cancer Institute, a lot of people are familiar with the Cancer Genome Atlas. There was another initiative that got launched at the very same time, and that was to go after proteins. The only catch is that we did not go after biology. We were kind of forbidden by the board because of issues that happened within the scientific literature at the time. So they brought me on board, and the idea was, because I had a background at that time in developing standards for the clinical community, developing round-robin studies, I brought a lot of rigor and reproducibility to the measurements that were used to do high-throughput protein-based measurements. We demonstrated those capabilities back to the NCI leadership, and subsequently then we started going after TGA samples. And once clearly you demonstrate, you identify new biology or help clarify questions that get arisen from genomic or other molecular characterization techniques, you begin to see the value from a scientific argument. So today, one of the things that makes CPTAC very unique is that for every sample that we obtain from a patient, we simply don't do genomics or we don't do proteomics. Each one of those will undergo DNA, RNA, and protein-based measurements. And all that information, we have computational sites that will integrate that complex information and try to put it out, and we give away all the content to the public. That's actually on the right-hand side of that slide. So the current iteration, we carved it into two areas. There's one that we call a characterization program. This is the one that develops those foundational databases that everybody loves to go after. So here we collect tumors. These are all going to be treatment naive. We compare it to the normal adjacent tissue. Once you characterize it as best as you can, we make all the molecular data available to the public. But the other thing we wanted to do to accelerate that initiative, which is really my heart's always been in, we have a translational program. That's sort of a high bar, because here we fund academic laboratories and their goal is to be conducting preclinical research. But at the end of those grants, hopefully, if the preclinical research is meritorious, they've developed this clinical question in mind and they will gain access to a clinical trial. to the samples from those clinical trials, try to further provide clarity to it. So with that in mind, the question is, what has this program achieved? And again, this is a very brief overview. So this represents what now we've been able to do in the past 7 years when it comes to the characterization of treatment-naive samples. We now have fully characterized 12 distinct cancer types. And if your math is good, you know, from the genomic side, it's a ballpark at about 24 to 25. But keep in mind, this takes time. We do genomic characterization with our colleagues at TCGA. That gets done by them. We, in turn, we add the protein information. And what these represent are what we call a flagship study. That's the very first time we're given a cancer type. We try to characterize it as best as we can. We try to interpret all the data, and then we release all the content to the public. Behind each one of these, there's typically about 50 additional papers that we kind of tease out the biology, but these are the main flagship studies. The other thing, because now we've gone through 12, we're in a position to do what a lot of colleagues in the genomic space does. Pan-can studies. It's always fantastic to compare what's unique and, you know, with the similarities, the differences between other cancer types. We're now following the same path. Now in 2023, we released the very first 5 pan-cancer studies, and we anticipate either by the end of this calendar year, by 1Q or 2Q of next calendar year, there'll be approximately 4 to 5 more pan-cancer studies. Again, following the path of the genomics community. But what about those translational sites? Gaining access to clinical trial samples is a very high bar because ultimately if those trials are going to be sponsored by the NCI, you got to go through a core correlative science committee, which does take a lot of time. You got to have absolute certainty in what you're trying to go after. We're starting to become quite successful now. In the past 5 years, this is not what we've been able to do. Specifically, we launched it going after 3 distinct cancer types that are shown there. Breast cancer along with ovarian cancer and leukemia. The leukemia, to be very transparent, that trial's beat AML, which is the Leukemia Lymphoma Foundation, but nevertheless, 2 out of those 3, we've already been able to tap clinical trial samples, adding greater value to what those trials are actually trying to do. And for the ovarian cancer, that's now in the process of hopefully being approved to gain access to their clinical trials. But then the other thing that we did 2 years ago, the leadership of NCI said, you know, it's kind of interesting. You're funding these sites, but you're kind of limited in a way to the cancer types you can actually look at. Because if you fund 3, you fund 5, that's going to be your limit. What if you sort of open up your sites to outside groups? And these outside groups potentially then could have additional cancer types. And if they're only performing, like the vast majority, genomic characterization, could these sites, with their expertise, some of our laboratories are now CLIA certified, would be conducting protein-based measurements on their samples. Forge these collaboration. Let's see what comes out of it and pilot this. Well, the very first 2 are listed on the right-hand side of that slide. So one of this is actually with AstraZeneca, and then the other one is actually done through a foundation. But now you could see there are outside groups we started to recognize that they would like to have their samples characterized, not just DNA and RNA that they were doing, but layering the protein information directly on top. But now this is where it comes to inclusivity of collaboration. I think one of the great successes that we're starting to see is with CPTN. And this started approximately about 6 years ago. So now published 4 years ago, there was an initial study that we were approached by colleagues and they said, we have this program, CPTN, it undergoes genomic characterization. We'd love to do something within this space. And the one that we actually looked at was pediatric brain cancer, GBM. I'm not going to go through all the details, but suffice to say, CPTN, the beauty of this relationship is already the material is genomically characterized. We, in turn, then farm that out to our sites. We perform the protein characterization. Our data analysis centers will integrate the data. But once that data was actually in hand, Adam and along with his colleagues who run clinical trials then looked at the data, and the beauty of it, it actually then provided critical support for the launching of a brand new clinical trial that's shown on the right-hand side that's not currently been done at the Children's Hospital of Philadelphia. And if you'd like to learn a lot more about it, what a lead investigator there is Cassie Klein. But this has now led to sort of acknowledgement. If this is not all would pan out like this, but if this is a glimpse of what you can get through collaboration, and by being inclusive when it comes to molecular characterization, because there's really no reason you cannot do all 3 types today based on platforms, can we expand this? And the answer is yes. So while the initial one here that's shown here, it's a combination between the CPTAC program, between CBTN, and the Kids First program that's funded out of the NIH Office of the Director, we're not going to be expanding these. And they're listed on this slide at a real big high level. With the Kids First, we're going to be expanding that collaboration. We're going to go after additional brain cancer types, looking at multiple subtypes. We're also going to be looking at blood cancer, ALL, along with TALL. Also out of the National Institutes of Health, NCI, there's a Childhood Cancer Data Initiative, or CCDI. That's another collaborative project that now we're going to be partnering with them on. And we'll be looking at about 1,000 samples from that initiative. There's another one that we're still trying to work out the kinks. I was really glad that I was able to meet the investigators here from the Linda Light Collective Foundation, but that's another one that we're going to be working on that focuses on a very specific type of a brain cancer, low-grade glioma. And one of the ones that I have to admit that I'm really excited about, and in fact, Adam was one that actually brought this one to bear, is these N of 1 based studies. I mean, these are individuals that they've exhausted every potential treatment that's out there, and they're looking for a lot of hope. So one of the questions is, do you have the capabilities, if we were to give you a sample, turn around not just the characterization, but the analysis, your interpretation of the data within a 2-3 week window? And the reality is that capability exists today. So we would like to do it. It's a learning process. We're going to learn how to put the data together, how to present it back to a tumor molecular board to potentially help drive the care for that individual. Personally, the way I see it is that the protein information, while it will not likely drive the actual care, because I think it's way in the early stages, I absolutely see it as providing clarity again, where you have questions that are arisen through genomic characterization, and this could actually help move things forward. Now, as I indicated, the other beauty of the CPTN and also CPTAC is one of our mandates. It's all that characterization data is to be made available to the public. So here's my cartoon that I always love to give. All of the protein data we developed for the NCI, the Proteomic Data Commons. If people are familiar with this, Warren Kibbe was out here, and I'm sure he probably talked about the whole infrastructure within the Cancer Research Data Commons, and there's different pillars. The Proteomic Data Commons is the one that houses all of our content, and it easily links out to our genomics data, which is housed within the GDC, and all the images that we also obtain from the pathology suite and also from the radiological side that's housed within the Imaging Data Commons. The other thing that we also give away, to be very transparent, we run not just comprehensive measurements, we could run targeted panels as you typically do within the genomic space. All the SOPs and every reagent that we develop is actually given back to the public. That's shown on that slide. And the last thing that I'll do is this one slide. So this is sort of a selfish pitch, but I thought I'd mention it here because I Adam's also going to be in Philly with me at that time. I actually believe that we're coming to a stage from a technology perspective. We have the ability to be performing this more and more, and it's becoming more common. So if from a technology perspective that is true, then what are the non-scientific forces that there's a lot of hesitation for people implementing this? And there's a variety of reasons. So myself, along with colleagues, we're going to co-chair a town hall session. And you can see the very distinct panel that we put on the right-hand side of that slide. And the idea is not just to identify what those non-scientific barriers are, but to potentially then identify how to circumvent them, to move forward. And hopefully what we'd like to get out of this town hall session is to develop a white paper and publish that back to the community at large. So with that, I'd like to say again, thank you very much. And I'll be happy to take questions at this point. Brian?
Thank you so much, Henry.